It's my pleasure to introduce Professor David Shea from Stanford University. Professor Shea is one of the most influential information theorists with significant contributions, particularly on fundamental limits of wireless communication. His vision on this topic has been reflected in his popular textbook, Thought All Around the Globe. David received his PhD from MIT in 1994. After that, he spent 1 year here at Bell Labs as a postdoc. During that time, he wrote an award-winning paper on capacity of wireless multiple access channel. After that, he joined the UC Berkeley as a faculty member. 2 years ago, he moved to Stanford. David received some of the most prestigious awards from not only from the information theory community, but also from all related communities, including communication, signal processing, networking, applied probability, and so on. More recently, he has been applying information theory to problems in biology. I spent 2 years as a postdoc with David. His passion in research, choice of problems, and clarity of thinking has had a deep impact on my research. With that, please join me in welcoming David.
Thank you very much, Mohammed, for the kind invitation. Thank you very much for inviting me back here. So I spent 1 year at Bell Labs, but that 1 year was very important for me, for my career. So thank you very much. OK, so today my title is Unreasonable Effectiveness of Information Theory. This title was stolen from Eugene Wigner's famous paper on the unreasonable effectiveness of mathematics in the natural sciences. But I thought it was very apt to explain some phenomena observed in information theory, which I want to share with you today. So this conference is about history, so start with a little bit of history. As we know, in 1948, Claude Shannon wrote his paper in which he invented a mathematical theory of communication. 70 years later, all communication systems are designed based on the principles of that theory. So to me, that is an extreme example of an unreasonable effectiveness of theory, right? How can an abstract mathematical theory have such a wide impact on the real world? However, today I want to dig a little bit deeper, and I want to talk about the unreasonable effectiveness of this particular theory. And the observation, which has been made before, is the following. Shannon looked at the problem of information limits without computational complexity consideration at all. Yet, 60 years later, after a lot of research, which was described on these 2 days of conference, ultimately revealed computationally efficient coding schemes that achieve these limits. So that, to me, is sort of in the core of the matter, the unreasonable effectiveness. That is, Shannon never considered computational issue. So why would there be solutions that are computationally effective? So to understand that a little bit better, we bring out our two heroes of the information age. We have Claude Shannon on the left and Turing on the right. So yesterday, there was some discussion about the meeting at Bell Labs. They spent a month actually in the same place, and they had lunch together and so forth. So unfortunately, we had no record of that conversation between them. However, we can simulate that conversation here. So Shannon would be telling Turing that, hey, you know what is the information limit for communication? OK? So that's the question I'm interested in. Turing, OK, with some modern interpretation, perhaps, says, well, actually, I figured out that optimal decoding of general codes is NP-hard. In fact, Alvin Berlekamp was here yesterday. He was one of the person who proved this kind of result, and there were a few other papers as well. That's true. However, in this conversation, Shannon had the last word. He said, well, NP-hardness is about the worst case, but I only care about the problem instances that matter. And the instances that matter are the ones which are information feasible. So I would like to take this point of view and ask whether we can use this information as a filtering of problem instances point of view as a guiding design principle for other problems. So this conference is about history, but it's also about the future. And so I'm going to talk about the future as in a problem that I'm working on right now. OK? All right, so this is what I've been interested in the past few years, which is high-throughput sequencing. So in this room, we're Bell Labs. Our main focus in the past 15 years have been on communication, wireless communication in particular. But in the parallel universe, in biology and medicine, something else is happening. And this is a technology called high-throughput sequencing. It started with the sequencing of the human genome in around 2000. 15 years later, the cost of sequencing has gone down 6 to 7 orders of magnitude. Right now, it's basically one of the central technology in investigations in biology and medicine. Okay? And as you can see, the price has gone down even faster than Moore's law. So I've been quite excited about this technology, and I've been thinking about various problems around this technology. If you think about this technology, basically there's this equation. So someone told me that this is a celebration of Shannon, and therefore there has to be an equation. And this is my only equation. My only equation in this whole talk, OK? And the equation is important. Sequencing is biochemistry plus computation. I have to admit, I know nothing about biochemistry. So luckily for you, my focus on computation. OK? So computation, there are lots of computational problems with high-throughput sequencing. But there's one very basic one, and this is the first one that essentially people have worked on. So I want to talk to you about my investigation on this problem in the past few years from an information point of view. So the DNA assembly problem can be explained very simply. And this is the problem. You start with a sequence, the genome, of length G. You pass it through a sequencing machine. This is the biochemistry. Due to various limitations of the biochemistry, it cannot read the whole string from beginning to end. However, what it can do is to generate n randomly located reads, which are short subsequences of length L. Length L in this example is 5. Now, the computation part comes, because I need to assemble them together. This is a computational problem to reconstruct my sequence S. OK. So I have my sequence S hat here, which is the output of the assembler. This is it. This is the high throughput. This is the assembly problem. I start with a genome. I get fragments, n of them of length L. We're engineers, so let's get a sense of orders of magnitude. OK. So G, typically of the order of 10 to the 6 to 10 to the 10. N of the order of 10 to the 7 to 10 to the 9. And the read length is about 100 to 10,000. So this is the scale of the problem. It's a pretty large-scale computational problem. We're talking about hundreds of millions or billions of reads here. OK? All right. So what's the theory available for assembly? OK, so we bring out Shannon and Turing again. So assembly is in the area of computational biology. The primary focus is on computation. In other words, Turing's descendants. Turing's descendant look at this problem from a computational point of view. And typically, it would formulate the assembly as a combinatorial optimization problem. OK? So this combinatorial optimization problem, and there are many versions of such problem, but one, simple to explain one, is called the shortest common superstring. So you have a bunch of subsequences, short. And now you want to look for the shortest sequence, long sequence, that contains the subsequence. Unfortunately, this and most other formulation of this problem is NP-hard. So NP-hard is one good thing, because it can allow you to be lazy. You can say, well, OK, it's NP-hard. And so let's just apply some heuristics, get some solution, try it out on some data, and hopefully it works well. So we can look at the problem from a different point of view in the past few years. We are from the Shannon School. So we start from the point of view of information rather than computation. Okay? So basic question here is the following. How much data is needed to unambiguously reconstruct the sequence? That is a very natural question asked by the information theorists, but not a very natural question, perhaps, for the other school. All right. So can answering this question, and this is the bottom line here, can answering this question not only give you a nice answer, but can help you to avoid the NP-hardness of the formulation. So the central issue about assembly are repeats. Think of assembly as a jigsaw puzzle, okay? So we have a jigsaw puzzle on the left. Everyone knows it's a hard jigsaw puzzle because there are a lot of repeats. So if you get a small fragment here of the jigsaw puzzle, you try to put them back together, you don't really know which piece where it belongs to because there's so many similar parts. On the other hand, on the right-hand side, we have an easier jigsaw puzzle where there are very few repeats and therefore much easier to assemble. So the fundamental limits of this problem depends on repeats. But how exactly do it depend on repeats? And that's the question we asked 3 years ago with my students. So the way we ask the question is the following. We extract a bunch of repeat statistics for any given genome sequence. This repeat statistics could be, back please. Repeat statistics could be that I measure for each length, repeat length, how many repeats are there in the genome, okay? And what we did was we derived a lower bound to what is possible, given repeat statistics of the genome to be assembled. And so I want to explain a little bit about this plot, which is the central part of the talk. Here we have on the x-axis, we have the read length, L. On the y-axis, we have n, the number of reads, the two important parameters of the problem. And we want to ask ourselves, how long the read length has to be and how many reads have to be in order for me to uniquely assemble this genome with this statistic, this repeat statistic. And this is the curve we derived, okay? Now, there's a famous line here called Landon-Waterman coverage. This was something known in the computational biology field. Eric Lander and Michael Waterman, two very famous people in this field, has derived the minimal number of reads needed in order to cover the sequence. So that is a low bound. If you don't cover the sequence, you can't even see parts of it. You can't assemble. However, what we showed is that the actual lower bound, better low bound, is this. Not only depends on the number of reads, but it also depends on how long the reads are. If I give you a read of one base pair or one symbol per read, no matter how many I give it to you, you can't assemble. Because the assembling is essentially based on combining reads together based on the overlap information. So the information is in the overlap. So this is our lower bound, which we derived. Now the question is, does this lower bound, this information limit, help me to design an efficient algorithm to get close to this lower bound? And that's what I want to talk about. But before I talk about that, we need to investigate, just like Shannon, we need to investigate what this limit comes from. So why is there a lower bound? The lower bound comes from repeats. And here's an example. I have this repeat, so-called interleave repeat, where I have a repeat here and a repeat here. And now a bunch of reads that are sampled from the genome, okay? And let me assume that the reads do not bridge the 4 copies of the repeat here. In other words, there's no read that goes from one side to another side of a copy, okay? I claim that given the situation, you cannot uniquely reconstruct the genome. And why is that? Well, if you look at this part of the genome and the associated read, the green part, and you look at the purple part, okay, which is between here, right? Now, these reads, they don't touch the blue parts, and these reads don't touch the blue part. So if I flip things around, okay, then there's no way you can tell the difference. In fact, the likelihood of observing these reads are identical, okay? So therefore, there's inherent ambiguity, and so we can prove the following result, which is a necessary condition for reconstruction is that all the interleaved repeats on this genome must be bridged by the reads. Similarly, you can prove an analogous result for another type of repeats, which are called triple repeats, where a copy appears 3 times. And here you can see you can apply exactly the same trick, which is you swap the orange and the green and the purple, and you get ambiguity. It turns out that these two are the types of repeats that really matter, and that computing based on these 2 necessary conditions gives us the lower bound. Okay, so now I want to ask, which is, how do I use the insight of the information limit to approach the limit efficiently? Okay, and this is joint work with my colleague, Tom Corte at Berkeley, and our joint postdoc, Elijah Morani. So first, we construct a natural structure to represent the problem. And this structure is called a read overlap graph, which is a structure where everybody uses in this field. So the structure works as follows. Each node of the graph is a read, okay? Each edge of the graph represents the amount of overlap between the 2 reads. So for example, here we have 4 because there's overlap CGCA with CGCA here, etc. Okay? So what is a reconstruction? What is a reconstruction? A reconstruction is a path that visits every node in this graph. Okay? It's a path that visits every node in this graph. Now, this example, the red one is the path. So you take this read, you go here, you go here, you go here, you go here, and the output of this is the sequence, ACGC, ATTC, GCG. Okay? So this is a reconstruction, a possible reconstruction. And so a solution is that we want to visit every node at least once. But there may be many solutions. So a natural solution is to find one that maximizes the total overlap. And this is called the optimal generalized Hamiltonian path problem. Just like any other formulation assembly problem, guess it, this problem is NP hard. Thank you. This problem is NP hard, okay? So that's the bad news, all right? So how do we avoid this NP hardness issue, okay? So in practice, remember, we have n of the order of 100 million to a billion. We have order n squared possible edges. So this graph is huge. How do I search for a path on this graph? Okay? So what is the most natural approach that even my daughter, 4-year-old, can solve on this problem? Well, you take a piece, and you find another piece which has the largest overlap, and merge them together, and just keep on continuing and hope for the best. This is a greedy approach. In terms of this graph, what we're doing is very simple. What we're doing is that we should, for each node, just find the edge with the best overlap and just keep that edge. So the name of the game is really to try to how to prune this graph so that it becomes manageable. And greedy is the extreme. You only keep the edge with the largest overlap. Okay? So here's an example where all the green edges are the only ones you're left with the greedy. Okay? So the question is this good? Does this solve the problem? Does this get to the information limit? So, well, what happens if there are long repeats? So let's look at an example. Here we have a repeat yellow of the genome, okay? And if we have this long repeat, let's see what happens, okay? Suppose we have a read, 2 reads, okay? 1 from each copy of the repeat. In the graph, these 2 reads are identical. So we merge into one node. Now, the solution of this problem, in other words, the real genome, I claim has to go through this node at least twice because it has to traverse both copies of the repeat. So that means there are 2 outgoing edges from this node in the true solution. And so therefore, a greedy solution, which only keeps 1 edge, will miss it. And therefore, in this case, the greedy approach fails. Okay? All right. So if you look at our information feasible region, what this says is that why greedy fail is basically whenever you have a repeat, the greedy approach would fail. Okay? And what this means is that you can achieve a certain region here. And I know that this is not equal to our lower bound. The reason is because the lower bound is because of the triple and the interleave repeat, not just simple repeats. So there's a gap here. And the question is this gap due to my lower bound being bad or the greedy approach being bad? And the answer is the latter. And how does information limit help me here? Okay? All right. Okay, so the greedy approach fails tells me that a true path must visit every node at least, need to visit a node more than once. Okay? But more than once could mean twice, 3, 4, 5 times. It doesn't really help me a lot. So let's ask the question. Does it need to visit a node more than 2 times? Strictly more than 2 times? So let's, again, bring out a situation like here, where a node, okay, is visited more than 2 times. So let's say 3 times. What does that mean? That means you have 3 copies of the repeat, 3 reads there, all merged into this 1 node here. Okay, so 3 times now. All right? So what's happening here? Well, it means that you have 2 loops, blue and red. Okay? Blue and red. But we know that corresponds exactly to triple repeat. And we know, because of low bound, that if this happens, that you cannot reconstruct. Because you can go from blue to the red, or red first and then blue. So this point actually lies in outside the information feasible region. So therefore, we don't have to worry about it. Information theory tells us that the complexity problem can be managed because the very complicated solution I don't have to care about. I can't reconstruct anyway. Okay, so there you go. So therefore, we constrain ourselves to visiting each node less than twice, and we use this insight to build an algorithm, what we call not so greedy, almost greedy, but not so greedy, where we only need to locally decide on 2 extensions, okay, only. Further pruning removes spurious edges, resulting in a sparse read overlap graph, okay? All right. So since this is a Shannon's conference, I figured we have to have 2 theorems. Here you go, 2 theorems. I don't have 13 theorems. Theorem number 1 says the following, that if all the triple repeats are bridged on the genome, then this algorithm returns a sparse graph with no spurious and no missing edges. That means no extra edges, and all edges that I need will be on the graph. Which means what? From a computational point of view, it means that the genome is actually a Eulerian path on the graph. Okay? Theorem 2 says that if furthermore there's no interleave repeats, okay, all interleave repeats are bridged, then there's a unique Eulerian path corresponding to the true solution. Okay? So this is greedy, and not so greedy is hitting very close to the low bound. Okay? There's a small gap here, which I can explain after the talk, but very small gap. And we run it on many, many genomes, and you can see the gap is very small in all the cases. So something interesting happening here. Let's recap. We start with the NP-hard problem, which is generalized Hamiltonian path on a read overlap graph. This problem in general is NP-hard. However, if we focus on the problem instances, which is information feasible, then what we showed is that we can reduce it to a sparse graph. We can locally throw away many, many edges, And all we have to do is to run a Eulerian algorithm on that, and that is linear time. So we solved an NP-hard problem in linear time by using information theory. OK? All right. So let me take 2 more minutes here if I have time. 2 more minutes. And I would like to say the following. We have solved a basic problem, the theory of a basic problem. Shannon tells us that once you solve a basic problem, you can add all the bells and whistles to solve more general problems, other problems, related problems. And one particular problem we worked on quite a bit is a so-called RNA-seq assembly problem. So it's very similar to DNA assembly, except now you have transcripts, which are sequences, and you have many of these transcripts. And again, you obtain reads randomly from each one of them. OK? So you obtain reads from many such sequences, and then you can reconstruct. OK? So on this problem, we again worked out the theory. It's an extension of the theory we talked about earlier to understand the fundamental limits of this problem. But for this problem, we did more. We did more. Because we figured out that doing all this theory is useless. Nobody listens to us. The only way we can get people in the field, that field, to listen to us is to build a software. And that's what we did. We built a software. We, of course, named that software. What else could we give it a name? Shannon. And the question is, does it work on real data? OK. Does it work on real data? We talked about all this theory, but what about the actual data? So just to show you that we are kind of serious about this, we actually did a lot of experimentation on real data. OK? So just to give you a scale of this problem, we have 135 million reads from a particular data set. This is human embryonic stem cells. OK? And what we did was we evaluated our software on this data set. And this data set has the advantage that it has a good ground truth from an alternative experiment. OK? All right. So here are the results. And we have our hero here, Shannon. Now he's reduced to software, so I don't know if he's going to be very happy about that. But nevertheless, he has no choice because, we just named the software Shannon. So here are the results. So Shannon, this measures how many transcripts can I recover. Shannon recovered almost 10,000 transcripts, about 20% more than all the other existing software. And if you separate out into category of the coverage of different transcripts, you'll see that Shannon wins in every category. And Shannon wins in every category not because it is very aggressive, because the false positive rate is also the lowest amount of software. So I hope that this evaluation and many others we did give some feeling that theory is not just theory. Following the Shannon tradition, we're pushing it to practice as well. So let me finish. Conclusion. Information theory is about fundamental limits. What I'm trying to say today is, hey, it is a constructive theory. It's not a theory about something abstract, because it tells you how to solve problems which are apparently NP-hard by essentially filtering out problem instances which are tractable. Thank you.
Thank you, David. We have time for one quick question. Okay.
Yes, there's some questions here. Don't clap yet. Don't clap yet. There's some questions there.
So my question is, with the type of measurements that they like to do or that they can economically do, where does it fit relative to the curves that you showed, the N and the L? Is it hard to get into that?
Yes.
Nice.
So right now, the dominant technology is called Illumina. The read length is about 100 or 200. It's way on the left of the fundamental limits. However, there are emerging technologies which are of the order of 10,000 read length, and now it's in the game. So we're actually building an assembler for that technology right now based on this theory.